Hello students, welcome to EPG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today we are going to talk on the types of lipid, which is a continuation from the previous lecture from the paper Lipid Metabolism. Today's objective of the paper is to know about the another type of complex lipid, which is called the prenol glycosides. We will discuss in detail how many types of prenol glycosides are present in a variety of organisms, and along with the prenol glycosides we are all discussing the significance of the lipids, especially which exist in association with the amino acids. So another important class, lipids containing amino acid, that we are going to discuss today, as well as the third type, which is another important type of lipids called as derived or precursor lipids. So we will also discuss about the derived lipids, its importance, as well as the significance of the derived or the precursor lipids. If we talk about the prenol glycosides, we will see that in Nicotiana, capsicum species and Lycium species, hydroxygeranyl nerol diterpenoid glycosides are the copious metabolites, which includes one or two acyclic geranyl linalool aglycones, which are conjugated to one or more sugar moieties. So we are discussing the different types of lipids, and these types include the simple lipids, complex lipid, and derived lipids. The simple lipids, including the fats, the oils, and the waxes, have already been explained in the previous modules. In the complex lipids, which includes the phospholipids, glycolipids, and proteolipids, has been discussed herein. Phospholipids are phosphorus containing lipids, whereas glycolipids are lipids containing saccharides or the carbohydrates. When lipids are amalgamated with proteins, it forms a complex mixture of lipids which is called as proteolipids. So in this module we are discussing about different types of glycolipids, and it has been continued from the previous modules. So the prenol glycosides, which is a complex type of lipids, it includes one or two acyclic geranyl linalool aglycones, which are conjugated to one or more sugar moieties. The chemical structure shown here has both R1 and R2 groups, and based on these R1 and R2 groups, the several analogues of the prenol glycosides have been isolated. Placopolyprenoside. It is a subtype of prenol glycoside, a complex lipid. It is a cytotoxic glycolipids poised with 35 carbon linear polyprenyl alcohol and a dixylosyl carbohydrate chain, which has been extorted from the Placotis simplex, which is a marine sponge. Plaxiloside, which is an analogous compound of placopolyprenoside, has a carbohydrate chain made up of 6 xylopyranose units as shown in the figure. Another important prenyl glycosides are glycosyl phosphopolyprenols, and these phosphopolyphenols of carbohydrate moiety act as coenzymes of membrane bound glycosyl transferases, which transfer glycosyl residues from the nucleoside diphosphate sugars to proteins, which act as acceptors. In prokaryotes, for example in bacteria, these glucosyl phosphopolyprenols are implicated in the genesis of complex polysaccharides of the cell wall, whereas in the eukaryotes they are involved in N-glycosylation process of the protein. An example structure of manosyl diphosphoicosaprenol is shown herein. Among the prenol glycosides, which is a complex subtype glycolipids, phenolic glycolipids or the mycosides also contributes to this important subtype. It comprises different structures such as glycopeptidolipids and phenol glycolipids, that is, the glycosides of phenol thiocyrrol dimycoserosate. An oligosaccharide moiety associated to a phenol thiocyrrol molecule is esterified by mycoserosic acids, hence establishes thiocyrrol. The mycoside family of complex phenolic glycolipids has the structure shown herewith. In Mycobacterium kansasii species, mycoside A, which contains 2,6-dideoxy 4-methyl Arab-2-methyl acetyl fuc-2-methyl Rab-2-methyl Rab. This specialized glycoside or prenol glycoside is present in the Mycobacterium kansasii species. Mycoside B, which is another very close isoform of mycoside A, is found in Mycobacterium tuberculosis, and this mycoside B contains 2,O-methyl rhamnose sugar, and hence other prenyl glycosides include methyl glucopyranose and two molecules of methyl rhamnopyranose. The phenolic glycolipids, especially from the Mycobacterium lepri, bears the inimitable determinant 3,6-dioxy-methyl B D-glucophosphonate 2,3-diomethyl L-rhamnose. It is a highly reactive and explicit for the sera from Lepromatous leprosy patient. Among the glycopeptidolipids, which are glycosylated fatty acylated peptides, rhizocaline, which is a two-headed sphingolipid linked to a galactose residue, has been established in a marine sponge named Rhizocalina incrusta. The structure of rhizocaline is shown at the right side of the slide, depicting the importance that this peptide with galactose residue possess antimicrobial and cytotoxic properties. Based on the important biological activity, few analogues, for example 2-ethyl carbamate of rhizocaline, were also revealed to possess anti-carcinogenic as well as pro-apoptotic properties. Another important glycopeptidolipids is Oceania P side, which has been described in Oceanapia philippensis, and this glycopeptidolipid bears the glycoside residue, which is glucose, linked at a different position, exhibiting antifungal activity, cytotoxic activity, or DNA damaging activity. The structure of Oceanapia is shown in this slide on the right hand side beneath. Mixotyrosides, an unusual glycopeptidolipids, bears rhamnose sugar moiety and have been isolated from the Myxococcus species with a normal or an isobranched fatty acid amide linked to a glycosylated tyrosine derived structure. This mixotyroside reveals a very fascinating structure surrounded by oxygen on the fourth side of the phenyl ring, and one of the important mixotyroside A is shown herein. Another important complex lipids with lipoamino acids, or lipids which exist in association with the amino acid. These lipoamino acids are actually the class of complex lipid with one amino acid linked or allied to long chain alcohol and acids, but it exists without the phosphate group. This lipoamino acid, a type of complex lipid, present solely in bacteria and lower plants, and these lower plants include algae, fern, and some of the protozoans. This lipoamino acid again can be divided into two important subtypes. The first is the lipids having an amino acid with N-acyl or ester linkages, and the second subtype is lipids having a glycerol with an amino acid with ether linkage. So the former subtype, which is with N-acyl or ester linkages, are again they are lysine containing lipids. Example is cyolipin A, or the ornithine containing lipids, and the example here is alpha N acyl oxyacyl ornithine. Another very important example is glycine containing lipids, which is N3D13 methyl tetra decanoyl oxy 15 methyl hexadecanoyl glycine. So the latter subtype, which is having glycerol with its ether linkage, is primarily derived from homoserine, but there are examples where you will find few with alanine moiety. The lipoamino acid, which is lipids with two important linkages, N-acyl or ester linkage or ether linkages. The N-acyl or ester linkages bearing lipids with an amino acids possess various kind of derivatives and are recognized as per their amino acid units. The lysine containing lipids, the ornithine containing lipids, and the glycine containing lipids. Example is cyolipin A, which is a lysine containing lipids, where the lysine is N-linked to a fatty acid, the R1 hydroxylated or normal, and to an ester-linked fatty alcohol, which is an R2, has been reported from the Streptomyces species. The ornithine containing lipids are those which is connected to the fatty acid R1 by an amide bond and by an ester bond to a long chain fatty alcohol, which is an at R2 position. The R1 chain has 16 to 18 carbon moieties, and the fatty alcohol have a cyclopropane ring. These type of lipids are found in photosynthetic purple bacteria, gram-negative bacteria, and in fewish genus of gram-positive bacteria. For example, Streptomyces and Mycobacterium species. But the ornithine containing lipids are absent in Eukarya and Archaea. The 3-hydroxy fatty acyl group is appended to an alpha amino group of ornithine, and a second fatty acyl group is ester linked to the 3-hydroxy position of the first fatty acid. The third, glycine containing lipids, have been identified in Cytophaga johnsoni, which is a gliding bacterium, and Cyclobacterium marinus, which is a seawater gram-negative bacterium. The glycine containing lipids contain 2 fatty acyl residues and an amino acid glycine by the acyl oxyacyl structure. In Cyclobacterium marinus, an iso 3-hydroxy fatty acyl group of N3D 13-methyl tetradecanoyl oxy 15-methyl hexadecanoyl glycine is connected by an amide to the glycine, and its hydroxyl group is esterified to a different iso fatty acids. The second group of lipoamino acid includes the ether linkage lipids having a glycerol with an amino acid, and they are primarily derived from homoserine, although few examples of alanine moiety also have been reported, which are characteristic of algae. These lipids are also known as betaine lipids because of the polar head derived from N trimethyl glycine, which is also called as betaine. In Ochromonas danica, which is a yellow-green algae, the homoserine derived lipids were first recognized, accounting for more than 50% of the total lipids, created by an ether linkage between the homoserine diacyl glycerol and are extensively strewn. During the phosphate stress in Rhodobacter spheroides and Cynorrhizobium millileuti, they were reported to be a replacement for phospholipids, and they induce the synthesis of diacylglycerol N-trimethyl homoserine. This has been well documented in Arabidopsis and oat, and the parallel interpretation were also reported for ocean phytoplankton using lipids of non-phosphorus nature and in retort to scarcity of phosphorus. This type of lipid was also reported from Acanthamoeba, which is a protozoa, Epidermophyton flocosum, which is a fungus, Chlamydomonas, Alva, Acetoploaria, Clostridium, Volvox, which are green algae, mushrooms, vascular cryptogamic plants, but absent in chlorella, tetraselmis, and seed plants. The R1-R2 acyl chains are 18 is to 0, 18 is to 2, where n is equal to minus 6, 18 is to 3, n is equal to minus 6, or n is minus 6 in Dunalilia, and 20 is to 5, n3 in Chlorella minutissima, and mainly 18 is to 1, n9 in Acanthamoeba. Since they have a quaternary ammonium group, the homoserine derived lipids look like phosphatidylcholine in some reference. In Acromonas danica, diacylglycerol, hydroxymethyl trimethyl beta-alanine, or the alanine derived lipids, was first recognized and observed to swap the homoserine derived lipids in brown algae, but are absent in the green. In Pavlova lutheri, diacylglycyl carboxyhydroxymethylcholine, which is another betaine lipid, was discovered. In Mycobacterium phyllae strain IST, a lysine containing diacylglycerol was isolated, which is esterified to 1,2-diglyceride by an ester link, and the foremost fatty acid substitution are tuberculosteric and palmitic acid. The structure of lysyl diacylglycerol is shown herein. Cerebrosides, also known as monoglycosyl ceramides. Cerebrosides are lipids containing ceramides, carbohydrate which is either glucose or galactose, fatty acid, sphingosine, but without phosphoric acid and glycerol. It is the common name for glycosphingolipid, an essential component in animal muscle and nerve cell membranes. A deficiency in deprivation of glucocerebrosides and galactocerebroside results in either Gawker's and or Graves' disease. A typical example structure of cerebroside is shown herein. Gangliosides. Gangliosides are also a complex type of lipids which contains the glycosphingolipid, and these glycosphingolipids are nothing but ceramides. They contain also saccharides like glucose or galactose with one or two more sialic acid content like N-acetylnuraminic acid that is linked on the sugar chain, which is composed of either glucose or galactose moiety. This N-acetylnuraminic acid creates the head groups of the gangliosides, which are anionic at pH 7, which is a neutral pH, and which discern them from the glophosides. In 1942, Ernst Klenk, who is a German scientist, gave the name of gangliosides, and this name has been bestowed to the class of lipids that were isolated from the ganglion cells of the brain. They are the constituent. More than 60 gangliosides are known of the cell plasma membrane. So most of them are involved in the modulation of cell signal transduction. So the gangliosides have been shown to be exceedingly vital molecules in immunology as well as in immune response. Natural as well as semi-synthetic gangliosides are considered probable therapeutic agent for neurodegenerative disorders. the example structure of these gangliosides is represented here. Another important class of lipid is cytolipids. These are the lipids containing fatty acids, sphingosine, glucose or galactose moiety. Continued with the types of the lipids, the most important, or rather a very important class of subtype, is the derived or the precursor lipid. The remnants, they are the substances which are produced as a result of the hydrolysis of simple or compound lipid. They are the precursors of the simple or complex lipids. Different types of derived lipids include alcohols, carotenoids, diglycerides, fatty acids, ketone bodies, monoglycerides, steroids, and terpenes as well. Since they are uncharged, for example acylglycerol, glycerides and cholesterol, these derived or precursor lipids are sometimes referred to as neutral lipids. Hydrocarbons, which includes the carotenoids as well as squalene, the precursor of the most of the sterols of the organisms. Cholesterol, for example, which includes cholesterol, coprostane, cholestanol, coprostanol, dehydrocholesterol, lanosterol, lathosterol, agnosterol, or even mycosterols, which are sterols of the yeast, ergosterol, the sterols of the fungus, phytosterols, the sterols present in plants, which includes stigmasterol and cytosterols. Bile acid, for example cholic acid, deoxycholic acid, lithocholic acid, glycocholic acid, taurocholic acid, and cholanic acid. These are some of the examples of the derived or the precursor lipids, or we can say subtypes of the derived or the precursor lipids. The most important part of the derived and the precursor lipids includes vitamins, especially the fat-soluble vitamins, vitamin D, which is also known as cholecalciferol, vitamin E, which is known as tocopherol, vitamin K, menadione. So, students, today we have learned about the prenyl glycosides, the important type of lipids, derived or the precursor lipids. So let us conclude today that we have discussed the types of lipids with amino acids, especially the prenyl glycosides, which is an important class of complex lipids present in most of the Mycobacterium species, and we have discussed the importance and significance of derived or precursor lipids, which includes vitamins, sterols, hydrocarbons, and so on.